Hello everyone. Today we're going to be talking about levels of protein structure. So in biochemistry, you generally talk primary, secondary, tertiary, and quaternary. But before I begin this video, I just wanted to remind you, thank you for saving my videos and liking them, but please also follow. Like, it takes time to make this content and I want to make sure that you guys are really enjoying the content. So if you want me to keep making videos, please follow me here and on YouTube. And let me get back to the video. So primary would be like a linear amino acid sequence. So it would be proline, glycine, serine, ... Like I chose randomly, but it's just you're gonna see like a line sequence. It's gonna be a linear sequence of amino acids. So primary, think of like number 1. So that's going to be 1 looks like a line. So just know it's a linear amino acid sequence. Then we go to secondary. In secondary, you have folding that's involved, right? So, but secondary, the main emphasis that I want you to think about is it's the backbone. It's not talking about the R groups, it's just talking about the amino acid backbone. So secondary, now that I think of it, it's an S. So just think of it how it's curving. Just think of the same way in the alpha helix. I don't know if that's going to help you, but if it does, great. But again, alpha helix is not the only type of secondary structure. You also have the beta sheets or the beta strands. So I'm going to talk about this in a separate video, but just for the shortness of time, this that I have in green can be parallel or it can be anti-parallel. Parallel that they're pointing to the same side, anti-parallel that they're going to the opposite side, and there's some other things to talk about in regards to that. But as of right now, just remember for secondary that it's mainly in regards to the backbone, the amino acid backbone. And again, it's alpha helix and beta strand, not like alpha strand and beta helix. That doesn't exist. It's alpha helix and beta strand. Okay, let's go to tertiary. So for tertiary, it's the 3D structure of an entire polypeptide. It's think about one chain, one complete entire chain. I'm going to go back to this example so you can understand. Quaternary is 2 or more polypeptide chains interacting. So for example, you have hemoglobin, right? So you have in hemoglobin, if you haven't learned it or if you did, great. It's 2 alpha chains, 2 beta chains. So it's not like just an alpha helix. No, like these, like I said, they're complete chains. And think of it as like you have 4 subunits in this 1 unit of hemoglobin. So each subunit or each chain, again, there's 2 alpha and 2 betas, would be an example of a tertiary, like 1 beta or 1 alpha chain is 1 tertiary 3D structure. But if you put all of these together like you are in hemoglobin and they're all interacting, that's why I kind of merged them together, then that is going to be a quaternary structure because it's 2 or more polypeptide chains interacting. Again, if you like this video, please follow and share, like, and follow here and on YouTube. Thanks again.